everyone. I'd love to thank the organizer for giving me an opportunity to present my work at London Health Sciences in Canada here. So I will be telling you a little bit about the work we're doing in testing of myeloid malignancies at London Health Sciences. The title of the talk today is Clinical Impact of Frontline NGS Testing in Patients with Suspected Myeloid Malignancy. Here's the disclaimer. So the objectives of the talk today are really to focus and introduce London Health Sciences molecular diagnostic program in the Verspeeten Clinical Genome Centre. It's always helpful to understand the context within which diagnostic implementation of the testing is being done. Then I'll tell you a bit about how we had implemented hematologic malignancy testing protocols at London Health Sciences. And we'll discuss the clinical validation of myeloid next-gen sequencing assay. After that, we'll focus on early performance data in the clinical lab setting and spend the rest of the talk really summarizing some interesting findings that come out of testing now over 3,500 patients with Tier 1 NGS-based testing for myeloid malignancy. So London Health Sciences Molecular Diagnostic Labs and Verspeeten Clinical Genome Centre. We are an integrated clinical diagnostic program as well as a translational research lab. We have about 70 or so clinical research and bioinformatic staff and students. We perform about 30,000 clinical molecular diagnostic tests and reports per year. They span a range of testing, including hereditary genetic testing to somatic oncology work. We are a province of Ontario reference lab for various different panel tests, including Center for the Ontario Epilepsy Genetic Testing Program. We do hereditary cancer panel testing, neuropathy and Charcot-Marie-Tooth testing, mitochondrial genome sequencing, lysosomal storage disorders, urea cycle disorders, to name a few. In Ontario, this testing is performed in centralized fashion, so single lab performs testing for the rest of the province. Whereas hematologic oncology solid tumor testing in particular is done on a regional basis. For hematologic oncology testing, which I'm going to talk about today, we are a reference center for approximately about 2.5 million people catchment in southwestern Ontario. We also do kind of global genomic profiling tests, including things like exomes, and we are a lab that's developed and recently implemented even clinical epigenomic testing in rare disorder. I won't be telling you too much about that today. I'll mention a few things at the end of the talk as to how it might apply to hematologic malignancy testing. So back in 2015, we've put together a working group that focused on trying to develop a strategy and plan for expanding and implementing more advanced testing, molecular testing for hematologic malignancies. We had really initially focused on, kind of primary indications of acute myeloid leukemia, lymphocytic leukemia, MPN, lymphoproliferative neoplasms, bone marrow transplants, and related tests. We consulted with the recommendations from the regional working group, including hematology, oncology, pathology, flow cytometry, molecular genetics, and we heavily relied on WHO guidelines. When we thought about putting together, molecular diagnostic testing or expanding molecular diagnostic testing, we really looked at the context within which this testing was performed in our institution. We are, as I said, a regional reference laboratory. We receive testing from our local departments within our hospital, but also from external hospitals as well. These samples usually reach the specimen receiving area, and then diagnostic triage happens. So in this option one, which we refer to as targeted molecular triage flow chart, generally what happens with samples is they would receive flow cytometry and special hematology testing, as well as cytogenetic testing. And depending on the findings in the testing cascade here, samples would be reflexed off to molecular diagnostic testing, where normally or historically we would perform RNA or DNA stabilization until we wait for the results for the primary testing in flow and special hematology and cytogenetics. And then we would decide whether or not to discontinue test or follow up with testing, molecular testing. And historically, we would have performed primarily single gene, single analyte targeted genotyping and real-time PCR type testing for a few indications. However, the alternate strategy at that time, back in 2015, was proposed was to look at using primary NGS panel, next-gen sequencing panel, instead of this kind of targeted triage for molecular diagnostics. And essentially, the workflow would be the same, other than every specimen would also, in addition to flow cytometry and cytogenetics, receive molecular diagnostic workup using next-gen sequencing panel. And we're looking actually not to use this as a reflex test, but essentially for every patient that suspected myeloid malignancy, we would perform an NGS test. And after a significant amount of consultation and discussion, we decided that we wanted to implement the option 2 at Lona Health Sciences. This is way back 2015-16 at the time we were discussing this. So at the time, we looked around to see what might be the most effective option to implement this. We need to make sure that the NGS assay was able to detect all of the common myeloid malignancy-related mutations. As I said, we consulted WHO guidelines for what genes are required, testing the DNA, but also specifically, we wanted to make sure that we're able to assess for DNA fusions as well. And at the time, we'd identified an assay that we wanted to work with and ultimately decided to implement it. So when we decided to implement it, we performed some initial validations that were highly technical in nature. So we identified a cohort of about 21 samples that included cell lines and manufactured controls and some specific patient samples with known variants. When we assessed these samples, as I said, we knew a priori what specific mutations to look for. We looked for a range of mutations from point mutations to tandem duplications and deletions and more complex variants like fusions and so on. And we had determined the high level of specificity compared to the previous data, and then high level of sensitivity. So we had about 95% sensitivity of 100% specificity based on those 63 mutations, about 29 fusions we looked at. And it turns out that for the 3 variants that were initially missed, resulting 95% sensitivity, they were actually present in the NGS reads, but were filtered out as a result of the filtering setup on the software at the time that was designed to filter out variants below 10%, which we've adjusted since. And I can say that at the moment, we're able to effectively call variants above 5% and above, and for specific mutations, even a bit less than that. So effectively, since the DNA specificity was complete, we also did a technical validation involving a number of other laboratories within the province to look at the concordance for calling all genomic variants in about 8 samples. And I say all genomic, all SNPs, etc., about 350 genome position. And that received relatively good concordance. This is back in the day where the assay was just beginning to be performed. So we're getting roughly about 95 to 98% concordance between calling, which we felt at the time was certainly sufficient evidence to proceed with the implementation of the testing. I should also say, based on some additional data, we demonstrated gene fusions were validated to sensitivity about 1%. We're actually able to go down to about 0.1%, but we set up 1% as a cutoff. And then DNA assays are validated with detection sensitivity down to about 5%. We also wanted to make sure that we had targeted assays available. And the reason why we wanted to have targeted assays available beyond the NGS panel, as I said, NGS panel was going to get implemented as a standard tier 1 test. There is a turnaround time, certainly requirement. We currently try to turn around NGS testing routinely about 2 to 3 weeks, which is sufficient. But occasionally scenarios when we needed to get much quicker turnaround, sometimes in a couple of days, for which we have specific targeted assays. So we validate a number of targeted assays, targeted DNA-based assays in the lab, and I've listed some of those here. Also, we validated a number of different fusion assays from RNA, and these are really designed to do things like minimal residual disease monitoring, and those are highly sensitive. They use real-time PCR, and they're able to go down to roughly log 4 or 5. For some of these, we actually use them somewhat routinely for some patients every 3 months and so on. So in addition to the NGS panel, we have basically a set of specific targeted assays that are used at clinician discretion, on occasion. So once we've implemented this, we wanted to see the impact of this implementation. So it was really exciting at the time because we were the first lab in the province of Ontario, and I think globally at the time, the first lab that actually attempted in clinical setting to use NGS-based testing, panel-based testing for myeloid malignancies as part of this tertiary, clinical center. We had some really exciting findings based on the first 380 patient testing. We published this in Hematology, European Journal of Hematology, demonstrating that about 50% patients had identified reportable variants, tier 1, 2 reportable variants, which was very interesting. Also, I should mention about half of the patients did not have variants reported. It's likely because vast majority of those patients did not have hematologic malignancy. Remember, we are testing all patients that are referred to our hematology program for workup for suspicion of a myeloid malignancy, and in this case, about half of the patients had received, reports with specified mutations. So when you start implementing NGS-based testing as a tier 1 or routine test, you have to consider building out the capability for streamlined data analysis, interpretation, reporting for these variants. At the time, of course, there's not a lot of commercial options that were, I would say, highly effective for this process. So we built in capacity internally, developed databases and tools for reporting. They involve currently a team of genome analysts, molecular geneticists, and pathologists who assist in review and assessment of these variants. Over the years, we've developed both gene and variant-specific reporting databases that are routinely updated through our internal team. For the clinical evidence and so on. We are routinely reporting tier 1 and 2 ASCO-AMP recommended variants, so they're clinically actionable based on the fact that they're either diagnostic, prognostic, or have an impact on clinical management. I'm just showing as an example there of a type of report that gets issued with every variant, so they normally reflect evidence for the pathogenicity of that variant based both on the publicly available databases, now also our internal database, as well as functional evidence in addition to any evidence for diagnostic, prognostic, or clinical management impact. We don't make specific recommendations on clinical management, but we do provide evidence that can be used for those clinical management decisions by physicians that are receiving these results. So I'll kind of fast forward a bit and focus on another larger paper we've recently summarized and published, and that focused now on about 2,000 or so patients that are tested in the first couple of years of testing. We really wanted to focus on a clinical utility and impact of implementing this testing. Out of at that time 2,023 patients were tested in a period of 2 years between January 2018 and December 2019. Out of those, 211 patients had no clinical details available within our internal databases. Amongst those, about 47% tested positive for a variant. 199 patients were diagnosed with lymphoid malignancies, so non-myeloid malignancies. We don't routinely use this panel for testing on non-myeloid indications, but occasionally physicians may request it and for specific lymphoid malignancy subtypes. And as you can see here, there has been evidence of a significant yield under certain scenarios. But as I said, it's not currently routinely used for lymphoid malignancies because the panel itself is not designed to cover comprehensively all the required molecular assays that would be important to test routinely with the panel. With the hope that in the future there will be an ability to kind of build out that capacity as well. So the next few slides are going to focus on about 1,613 patients within the stream of suspected myeloid malignancy and what specific findings we got there. So overall diagnostic yield, it stayed relatively similar to the first few hundred patients we tested. So we had amongst about 1,600 patients, there were about 700, so about 43% with a positive tier 1, tier 2 reportable variant. about 58 patients, or about 4% were shown to have a fusion detected. And I would say that this was also across the age group. So we've split the patient cohorts to adult and pediatric population. As you can see, in pediatric population, we've seen about 30% diagnostic yield for tier 1 to DNA variants, and about 11% for gene fusions. So the next question we asked was, well, what are the genes that are involved, in these specific clinical subtypes? Is there any particular enrichment? So we've split the gene groups based on their function. And one very common theme that's come up is that epigenetic regulators are quite commonly involved. So for MDS, for example, there's certainly a significant enrichment for epigenetic regulators, followed by genes that regulate splicing and signaling pathways. For MPN, signaling pathways and epigenetic regulators are also represented. And for AML, again, epigenetic regulators such as DNMT3A, IDH1, IDH2, TET2, and so on are enriched, followed by signaling pathways, transcription factors, and tumor suppressor genes. We are seeing significant enrichment of gene fusions, of course, in CML with BCR-ABL and AML. But this is an interesting thing to mention, is the evidence that many of the genes that are often affected are involved in epigenetics, so non-genetic mechanisms that are impacting these tumor subtypes, or hematologic malignancy subtypes. So then we wanted to ask, well, what's the specific prognostic related clinical utility for the findings? So in the cohort of individuals that were demonstrated to have acute myeloid leukemia, there's about 100 patients total within this cohort. 91% tested positive, out of which about 17 fusions were identified. This was interesting, and what's also interesting is about 71% of them had molecular markers that were categorized as per ELN 2017 risk stratification guidelines as either favorable, intermediate, or adverse effect. And about 34% had variants, other clinically relevant genes that didn't necessarily fit into those 3 specific categories. We did a comparison of these patients where we also had cytogenetic karyotype data, and amongst those, only about 25%, the ones that had the complex karyotype, we had certainly strong indication of adverse prognosis. Amongst 46% of patients with normal karyotype, 26 or 70% were positive for a prognostic marker identified by NGS. So certainly this was really convincing evidence that NGS profiling allowed for identification of prognostic markers beyond what's classically available to be able to determine by karyotyping alone. When we take a look at the MDS patients in this cohort, 160 out of 223, about 71% were positive by NGS. We looked at risk stratification markers, and based on the NCCN guidelines, and we found that SF3B1 mutation is a fairly common mutation identified here. It's an independent favorable prognosis marker, was detected in 20.7% of patients. 14.8% were negative for additional poor prognosis variants. So sometimes you actually have SF3B1 in combination with also a positive prognostic marker, likely indicative of progression of this tumor type. And 34.2 had an independently associated poor prognostic marker. So again, clinically useful information related to prognosis of these patients is being generated for a substantial proportion of patients with MDS. So looking at clinical utility of NGS and MDS patients and comparing it to the IPSS comparison. So IPSS scoring system was applied by the hematologists through hematology review to subclassify patients with the MDS. When we looked at correlation between these classifications and the NGS findings, we found, for example, that 51% of patients with IPSSR revised prognostic classification actually had, with a good prognostic classification as per IPSSR, had poor prognostic molecular markers identified by NGS screening. 3 of 3 with the intermediate prognostic classification by IPSS had a poor prognostic molecular marker, and about 61% with poor or very poor classification by IPSS also happen to have poor prognostic markers by NGS. SF3B1 variants were enriched in the good prognostic group as what would be expected, whereas P53 was enriched in the poor prognostic group. So again, this further emphasizes the additive utility of NGS beyond the standard scoring systems. In the cohort of patients that were classified by hematology as NDS-MPN, we again saw a very significant diagnostic yield, about 83%, and again, identification of both positive and negative prognostic markers. When we looked at specifically in patients with myeloproliferative neoplasms, we assessed about 379 patients with suspected MPN. 70.5% had an identified variant, and as expected, 49% were JAK2 mutations, 16% were CALR, and 5.3 had mutation MPL gene. 5 patients had JAK mutations other than the common V617F, and 87 JAK positive patients had variants in other additional genes, and there's a circular plot that shows some of these additional genes that had been detected. These additional variants identified by NGS beyond JAK2 mutation predict prognosis and leukemic transformation in these patients. We'll look at kind of within the whole data set, we can find that we're starting to see patterns that are specific for AML patients for the different subtypes of myeloid malignancies. So over time, we've seen that certain correlations between co-occurrence of variants exist that are dependent on the subtype of myeloid malignancies. You can see here depicted in the circles plot. And what's really interesting about it is now that there's certainly an evidence that, for example, triple mutated AML patients happen to have significantly inferior survival. So again, combination of these markers that can be detected by NGS parallel screening can provide additional information beyond assessing, single marker at a time as we would have historically done. The other question we want to ask was, what was the concordance of fusion detection by NGS with fission karyotype, which is, classic way that we currently do and historically had done this testing. So as I mentioned earlier, 58 patients, or 3.6 from this cohort, had gene fusions. Largely, there's 10 different types of fusions that we detected, many of which are BCR-ABL for CML. So 37 out of 38 patients that also happen to be tested by FISH for that specific, biomarker had been detected. So one patient was actually missed by FISH. 39 out of 45 that were detected by NGS were also detected by karyotype analysis. So 6 weren't detected. So it's relatively good concordance, and it also shows that NGS can pick up some fusions that may not be detectable either because of low frequency level or some other technical issues by classical techniques. I should also mention that 11 patients tested positive for fusions involving IgA gene by FISH, but not by NGS, mostly because this particular analyte is not present in the NGS panel. So we also, over the last 2 or 3 years, have had a chance to look back at the data and see if there's an opportunity for us to improve how we utilize testing. And in particular, is there an opportunity to remove redundant testing and quote unquote choosing wisely? And we determined that you can find a bit more detail in this reference publication that there are scenarios where there is redundancy between NGS and karyotyping. So for example, in cases where we have strong evidence for blasts and dysplasia, we normally now routinely perform NGS and cytogenetics simultaneously. For patients where there's no blasts but there's dysplasia evidence, we definitely do the NGS as a standard tier 1 screen. Whereas in patients with plasma cell neoplasms, there are less than 5% who are currently not recommended cytogenetic testing. And based on this triage protocol, of course, specimens are stabilized and they can still be requested by a physician if specifically required or indicated. But about 75% of those individuals that recommended to stop cytogenetic testing are ultimately canceled. So it ultimately allowed us to significantly reduce karyotype workload in patients where we don't find that to be useful additional information beyond standard protocol that now involves NGS. Elevated hemoglobin is a common reason for referral to hematology. So we wanted to look at this in this paper. We described the mutational analysis in patients with erythrocytosis, and as expected, we found that there's significant enrichment for individuals with JAK2 V617F mutation in exon 14. There's a couple of patients with exon 12 mutations as well, largely associated with polycythemia vera. And then we also identified additional individuals with variants. A bit more about it on this slide. So out of 58 individuals that tested positive for JAK2, 57 had the classic mutation, one had an exon 12 mutation. We also found the co-occurrence of additional mutations in this patient population. And as I said, some of them can be prognostic or indicative of progression. And additional non-JAK2 mutations potentially have prognostic relevance. As I said, out of 466 JAK2 negative patients, they were diagnosed with secondary erythrocytosis mutations that were found in 6%. We think that this likely reflects incidental age-associated clonal hematopoiesis of indeterminate potential. And finally, this is kind of my last slide here before the conclusions. So since the end of 2019, so between 2020 and May 2021, we've added another 1,600, so roughly at about 3,500 total now up until that point. I think at this point we've tested close to 4,500, by about 3,600 total at that point when we assessed the database. Out of the additional 1,649, we're seeing the similar diagnostic yield leveling out at about 42, 43% with tier 1, 2 variants. What's really interesting now is that as this database has increased, we're able to get additional inference about frequency of some of these tier 3 variants, so variants of unknown clinical significance, and evidence for enrichment in certain leukemic subtypes. So as we increase this database, it's proving itself helpful in the ability of reclassifying some of these variants towards tier 1, 2, or otherwise tier 4 variants. One of the things I just want to mention here at the end is that the epigenetic factor, right? So a lot of the genes that are involved in hematologic malignancies are related to involvement in DNA methylation, histone modification, chromatin remodeling through epigenetic pathways. There's been a lot of interest lately and evidence in development of epigenetic profiles associated with the underpinning clinical subtypes or genetic mutations. We've seen that work done in constitutional hereditary disorder. Our lab's done a lot of work in that space. There's evidence emerging in brain tumor subtypes, even some early evidence in hematologic malignancy. So our lab's very interested in this space. So in the coming couple of years, we are going to be doing kind of systematic assessment of epigenomes or DNA methylation profiles in this very large database to see if we can identify DNA methylation patterns associated with different acute leukemia, myeloid malignancy subtypes. And the goal for that would be to see if they can be used then to enable reclassification, for example, of significant percentage of these tier 3 variants. Tier 3 variants we don't report, but if we're able to determine clinical impact of these variants, it would certainly allow us to significantly increase the diagnostic reporting yield in these populations. So really exciting kind of work we're looking forward to doing in the next little while, and hope to share in the coming couple of years. And with that, I'm going to just conclude the presentation. So the key findings are that there's an increased diagnostic yield of NGS compared to classical diagnostic techniques across the myeloid malignancy subtypes. Well, we would have expected that, and certainly we're seeing that leveling at about 43% in this all-in population. When I say all-in population, it's about half of the patients that we screened routinely don't turn out to have hematologic malignancies, but we screen them nevertheless because it allows us to optimize the protocol and turn around these results relatively quickly without having to go through these triage procedures. NGS has an independent impact on clinical management. As we've seen, redundancy in genetic testing can be avoided by careful triaging, and I'm showing some examples of that. We demonstrate evidence of feasibility and potential clinical utility of integrating NGS first approach for patients with hematologic malignancies. And I also want to share really exciting news. As I said, we've implemented this NGS-based protocol for testing of patients with myeloid malignancies in our institution. We're the first lab in Ontario, I think amongst the first ones in the world to do this. And evidence that we've published over the years, but others as well, has ultimately contributed towards now in 2023, NGS panels being recommended as the primary screen and are endorsed and are about to be funded as a standard test through Ontario Health and Cancer Care Ontario for patients with indications of AML and myelodysplastic syndrome type indications. It's really exciting development. We're going to see a vast majority of patients with hematologic malignancy indications be standardly tested through these integrated DNA and gene fusion NGS panels going forward. And clearly, it's going to be really exciting to see what kind of impact we make on our patient populations. And with that, I'm just going to really thank you for your attention. I did a list here some of the publications that came out of this work over the last few years. I encourage you to take a look at it in a bit more detail if you have any questions. Thank you for your attention. Thank you for your attention.
I'd like to thank the organizers for the opportunity to present today at Oncomine World 2023 on the comparative performance evaluation of a commercialized laboratory-developed test in our laboratory, the GeneStrat NGS liquid biopsy test. This test is used for the detection of copy number amplifications, fusions, which includes exon skipping, somatic single nucleotide variants, and indels. Myself, I'm Gary Pestano. I'm the Chief Development Officer at Biodesics. I'm also the New York State Lab Director for our Boulder Laboratory here in Colorado. So thank you again to the organizers, and we'll jump right in. Disclosures: Thermo Fisher and its affiliates are not endorsing, recommending, or promoting any use of Thermo Fisher products. And myself, as a speaker, I was provided an honorarium for this presentation. So with that out of the way, I'd like to introduce Biodesics briefly. We, as a company, have two core business units. On the left is our biopharma partnering side of the business, which is really disease agnostic. And on the right is our clinical commercial laboratory testing business. Today, we will be focused on the GeneStrat NGS genomic test, which is targeted commercially on lung diseases. But as I'll share with you, the NGS test is really pan-cancer and was developed and validated in this way. The importance of having a liquid biopsy test is really illustrated by the rapid turnaround times that are afforded for these clinical tests. Why is it important to have rapid turnaround tests, and how does liquid biopsy testing fit in to rapid turnaround time? Well, in the case of lung cancer, which is where we're focused and which is where we've developed the GeneStrat NGS test to be targeted to, approximately 24% of non-small cell lung cancer patients will not have sufficient tissue for NGS testing to be performed, and that is at all. About further, 27% of non-small cell lung cancer patients harboring an actionable oncogenic mutation may begin treatment before receiving genomic results. The Biodesics GeneStrat NGS genomic test turnaround time, or TAT, averages about 98% of the time, delivery in 3 days or so on average, and this is recently as Q4 in 2022. By comparison, other commercialized tests on market, these are not inclusive of, for example, in-house hospital tests or by pathology departments, again, where information and specimens are readily available. Those can be as fast as a blood-based test if conducted in-house where everything is available, but generally for those send-out commercialized tests like the GeneStrat NGS test, turnaround times can vary 3 from 7 to 14 days or as much as 26 days when sent out. So what is the GeneStrat NGS genomic test? Well, with our focus on rapid turnaround, we turn to liquid biopsy to develop this test. It is a qualitative laboratory-developed test designed to aid physicians by providing molecular characterization of a cancer patient's disease. We developed this test and validated it using a proven clinical platform and technology. It tests for 52 well-characterized and prevalent cancer-related genes as an aid in treatment decisions. You can see the table on the right, which outlines the tumor types, for example, non-small cell lung cancer, colorectal cancer, endometrial, etc., and therapy recommended genes listed over on the very far right in the same table. The test averages 3 business days turnaround time for sample processing to results ready, and I showed you in the previous statement of the clinical need of how we're doing compared to this metric. Again, the test is intended for patients diagnosed with advanced stages of cancer, and in the case of Biodesix, our focus is on non-small cell lung cancer. The test also provides a report of genetic variation as well as references associated with the approved therapeutics, essential elements for the safe and effective use of the corresponding therapeutic products. So quite comprehensive, and this is illustrated in the next slide. Now a little more busy in terms of what classes of mutations and then what specific mutations are included in this 52 gene panel. First, starting with single nucleotide variants, or SNVs, insertion deletions, or indels, we consider these 2 different classes, but in terms of validation, may be approached very similarly, and I'll show you how we approach the validation of the targets in this mutation, or these 2 mutation classes. There are 44 SNV indels in the 52 gene panel. In terms of fusions and exon skipping variants, the gene targets are shown here, and then there are quite a few copy number amplifications that are also included in this panel. 12 fusions as well as 12 copy number amplifications to be exact. In terms of the performance of the test on market, there are certain limitations, and these are listed below in terms of the utility of the test, how it was validated, and can be used, and this appears on the test result report. A little bit about validating and developing a pan-cancer test. We actually use the same process for any of our molecular tests, all of which are New York State CLEP approved. So on the bottom, I provide references that include the New York State oncology guidelines, which if you're a test developer, you may consult. But very similar to this, I've outlined how we've adopted those CLEP guidelines, as well as relevant FDA guidance. And we have also published all of the validation data associated with our molecular tests. So this often starts with feasibility. So initial design, synthesis, depending on what your targets are, looking more at specificity versus sensitivity at this time. So we may employ synthetic targets, cell lines, well-characterized materials in the very early feasibility. Once we've got the beginnings of the assay, the next step is to then verify various parts of the process in establishing a clinical test workflow. So we look at pre-analytic, for example, ship of specimens, how are we going to package them, how are they going to be held in terms of temperature control. Analytic as well as post-analytic variables that could impact the results of those tests. Validation is then conducted in the case of the pan-cancer test for each variant class. So I'll take you through how we establish this for example, SNVs, indels, fusions, which includes exon skipping, as well as copy number amplifications in this test panel. So we started initially with establishing a lower limit of detection, or LOD, followed by precision, examples of robustness. So we conduct 21 days of consecutive testing, at which time we look at our standard deviations around passing or not before we are on market. Ship and shelf stability studies. Of course, you want to know shipping conditions. The GeneStrat NGS test, we advertise ship stability of up to 7 days. And for the NGS testing, we use a Streck tube. Many of you are familiar with the Streck tubes. In terms of shelf stability, this refers to in-process materials, how they're held, whether it's room temperature on ice or frozen. And then we continue with our normal healthy donor specificity, and I'll continue down this path and share examples. Clinical sensitivity and specificity is of critical importance as we go down the validation path with known reference specimens, as well as when we get to future studies. And I'll share with you how we determine accuracy using orthogonal technologies, for example, and or external clinical laboratory partners. We also conduct what is called a final acceptance test, or FAT, with field specimens before launch. This is a series of specimens that can be shipped around in the same conditions under which we would do commercial testing, and thereby test the entirety of our system, which includes pre-analytic, analytic, as well as post-analytic variable evaluation. And then finally, before commercializing a test, of course, the development of SOPs, standard operation procedures, work instructions for our clinical laboratory personnel, but as well for our support teams, so customer care teams, training and competence for all personnel, medical affairs, field training, as well in terms of talking to the test, educating physicians, as well as our regulatory submissions. In the case of our NGS test reimbursement, dossiers were also required, and this is also a necessary part of that test development. And then once on market, our quality system takes over, and those quality performance metrics on field as well as internal metrics are managed. So on to some data. So first, in terms of this pan-cancer panel, we divided our validation studies into two sections. So strategically, SNVs and indels are, if you will, more facile in terms of how they may be managed in terms of validation. Very similar kinds of mutations in these two classes. So in the box here where I've highlighted, I've got examples of SNVs, and if you're in the lung cancer space, you're familiar with some of the examples shown here, EGFR, for example, L858R, KRAS G12C, insertions and deletions as well in the EGFR gene, DEL19. So the way these experiments work, and I'll just share snippets for SNVs and indels and be much more detailed around fusions and CNAs, because I realize much less is known about those two mutation classes. So we'll get to those in a second, but I'll really provide summary of the published data around SNVs and indels. But we started out with cell-free total nucleic acids, or CFTNA, which were isolated, extracted, and then profiled. Again, the focus in terms of the workflow is looking at our turnaround time, and we published this work in January of 2021, where not only we validated for SNVs and indels, but also proved out the turnaround time of 3 business days in this workflow. So in summary, what we have done is validated and published on this highly sensitive amplicon-based targeted panel on a semi-automated platform. So I just wanted to introduce that what we're doing here is semi-automated. There's still several pieces of equipment in terms of library prep and then sequencing. So a little bit more hands-on, because as we get to what's next, I'm going to contrast what we do today for GeneStrat NGS with what is to come as well for us. So in result conclusion, you see here analytic and clinical specimens were analyzed with 95 to 100%, depending on the reference variants we detected. And that is shown on the next slides in terms of data. So in summary, for the clinical sensitivity and specificity, so again, if you refer to the publication, you can see all of the data around analytic performance, but I'm going to summarize this in terms of performance with clinical specimens. So we evaluated 53 clinical reference specimens for initial accuracy. Accuracy was done against a ddPCR, so the GeneStrat ddPCR test, reference specimens against the new GeneStrat NGS test. You can see the R-square values here really on the order of 0.99 with a high degree of concordance. BRAF V600E, if you're looking closely, we had 2 false negatives with the NGS test, but otherwise outstanding performance. So moving forward, as I said, I'll spend a little bit more time focused on copy number amplifications and fusions. Again, I've highlighted in the box the copy number variants, CNAs that are present in the panel, as well as examples of the fusions. In terms of performance, you can certainly read into this or get in touch with us after the paper is published. This one is in Diagnostics in 2022. Here, for the limit of detection, we established using remnant CFTNA again, admixed with cell line materials, replicates a single CNA and a single fusion. So that's what's shown in the initial sensitivity dilution series here on the top of the panels. First establish what that initial cutoff should be at the LOD. For fusions, that was established at an average of 42 copies. And for copy number amplifications, that was established at 1.4-fold amplification. We then selected 3 positive specimens for each of the CNAs, so different CNAs, and 3 fusions. Again, 3 different fusions, and that's shown on the bottom here where you see sensitivity verification, and looked at the cutoff around the limit of detection for detection or not. And we were successfully able to verify performance fusions at 42 copies for calling clinical specimens and copy number amplifications 1.4-fold for calling copy number amplifications or CNAs. So that was the first series of experiments. This is much more comprehensive now. Once we've established our limit of detection, we then looked at performance around precision. So how does this detection hold up? Again, we've done the same thing for SNVs and indels. I just didn't share all of that in the interest of time today. But here you can see the repeatability and the intermediate precision study results. Graph A shows repeatability, B interday, C inter-operator, and D inter-instrument variability. And the results really shown here in the bullets summarize the data. Overall hit rates for the CNAs and fusions were quite high, 100% with 18 instances of fusions tested, and 97% for 29 instances of CNAs tested. Average CVs for fusions ranged from 8% to 35%, and ranged for reproducibility and repeatability. And for CNAs, average CVs were in the order of 1.8% and ranged from 0% to 5.49% for repeatability and reproducibility, respectively. In terms of specificity, again, the same thing was done for SNVs and indels. We test against a series of normal healthy donors. So these are individuals that are drawn and their specimens put through the workflow. And here we're looking for false positives. And you can see, regardless of mutation class, none of the samples demonstrated here in this example, CNA or fusions being present. So that summarizes what we've done to validate the NGS tests for GeneStrat. Now I want to take you forward into what's next. And I think this is really, really compelling. So we've gone from the NGS GeneStrat test, which is currently commercial, and as I said, can be conducted within an average of 3 business days, on a semi-automated amplicon-based sequencing system, to now evaluating in the laboratory a fully automated NGS testing system, which is also amplicon-based. Different panel, different bioinformatics, different instrument, happens to be from the same manufacturer, of course, and looking at remnant specimens, so remnant clinical specimens, which have previously been tested for the 4 mutation classes using the semi-automated system. So on the left, I'll take you through a series of slides where we have the reference result, and then on the right, with the new fully automated system, where we've got the same specimens tested now, but with the different panel, 50 genes instead of 52, bioinformatic platform that is independent, and then, of course, for variant calling, and then on a different sequencer. Upshot is you can see in the green where we've highlighted the concordance. All samples were concordant at the label level, that is detected or not detected. But more important, you can see the molecular frequency, is really, really tight between the two platforms. And this is shown in the bar chart on the next graph. Current semi-automated system in blue, new fully automated system in green, and you can see the frequencies are really, really very consistent. We did the same thing looking at fusions and exon skipping with CNAs, as you can imagine. So on the top series of samples that were positive as reference specimens from the semi-automated system, and then tested with the fully automated system for those same CNAs. You can see the concordances again, 100% concordant. On the bottom, 2 examples of fusion, exon skipping, and you can see again on the left, the reference, on the right, the test with the new fully automated system, and again, 100% concordance. You look at bar charts, you can see that performance very, very similar as well between the two platforms. In summary, then, last 2 slides will take you through what we're continuing to do with the GeneStrat NGS test. 2 clinical studies to add to our clinical utility. First being an observational registry study, and the second being an independent sponsored study into which we're looking at tissue and NGS concordance. Both of these studies you can read about at clinicaltrials.gov. But again, test development, test validation on market, and we continue to do prospective study, clinical utility validation studies, even while on market, all necessary part of creating a quality product. So in summary, I think I've shared with you the development of a new liquid-based targeted amplicon semi-automated test, details around which you can find in 2 publications in 2021 and 22, the journal Diagnostics, the GeneStrat NGS test. We discussed data related to the detection of the 4 major mutation classes, SNVs, indels, fusions, and copy number amplifications. Accuracy was also demonstrated for the semi-automated, currently on market NGS test, and a new independent automated platform, and where we showed 100% concordance again for all 4 mutation classes using remnant clinical specimens. Clinical impact to decision-making in terms of turnaround time was demonstrated with on-market data for GeneStrat NGS, where we showed greater than 95% of GeneStrat NGS tests are ready for delivery in an average of 3 business days. And what we're all looking forward to is bringing on the new fully automated NGS test with its potential to deliver test results with even shorter turnaround time of less than 3 days to results ready. So I'd like to thank again the organizers for inviting me to do this and the greater Biodesics team. Thank you very much to our donors as well as our physicians.
Well, thank you very much again to Bekim and Gary for their presentation. Very insightful, a lot of useful information. In fact, I now welcome them back to join me for this Q&A session. Thank you very much, both of you. And we'll start now with a few questions. And the first one will be actually for Bekim. Bekim, once again, thanks for your presentation. The first question is about diffuse large B-cell lymphoma. How do you actually look for MRD in this specific subtype? What do you do in low lab?
So as we're talking about the NGS assay here, so we're currently not using NGS for MRD. Generally speaking, for most of our MRD assays, we use targeted real-time PCR-based techniques or other kind of targeted kits. But yeah, NGS we're not currently using for MRD. Part of the reason why is because it's not validated to go to the levels that we would really need. Yeah.
Any plan to maybe go and move forward, or in case should there be an NGS solution that fits your need? Would you say it is an opportunity, it is a possibility to use as well NGS, or is there anything that prevents you to do that?
Well, no, I mean, ideally, you'd want to be using NGS for MRD assays in general, because there are a lot of different targeted assays that would benefit from having MRD kind of level sensitivity. We, as I mentioned earlier, do a lot of targeted assays, including real-time PCR based assays for specific indications for follow-up testing. Having a single test to do all that simultaneously would be definitely something that would be very useful.
Thank you very much. Now, second question is for Gary. Gary, thanks for your presentation as well. Very insightful. A lot of very solid data that demonstrate the value of the GeneStrat test. The question is about, you mentioned some of the clinical trials that you are running. Can you share with us when the readouts of those trials is, or if there's any interim data, when you're going to plan to share something more around that? Thank you.
Yeah, no, great question. I believe one of those trials will read out next year, 2024. We are not the sponsor, so I'm not really at liberty to discuss interim data. But of course, we're all interested and excited about reporting on the liquid trial. That one's a concordance with liquid and tissue, so that'll be interesting and exciting. And then our study that we've sponsored, we'll release data as it comes mature. Luca.
Yeah. Thank you very much. Thank you very much for that, Gary. Now, going back to Bekim, another question, this time on multiple myeloma. In your opinion, how far away are we from NGS replacing FISH? And I mean, this is a recurrent question, I have to say. It was also coming up in the European session. And just to put it into context, in the solid tumor field, there's been now a lot of replacing FISH, with NGS because solidity of the assay. And in general, people feel very confident that they can do that. The question is, what about the heme? Will deals persist to stay, or how far away are we from this swap?
Well, I think certainly the trend is towards replacing FISH by NGS-based assays. When we talk about FISH, we're talking about being able to detect copy number alterations, fusions as well. In the context of fusions in particular, we have to worry about known versus the unknown, right? Being able to detect fusions that we're able to detect by targeted PCR assays versus assays that are allowing us to actually detect fusions that are more rare or novel. So I would say, in general sense, I think that the field is certainly going in that direction. I do expect that more and more FISH and or microarray-based testing is going to be replaced by NGS-based assays in oncology, as well as we're seeing in constitutional hereditary testing. The key benefit, of course, is that you're able to run these assays as kind of in a holistic sense for many different indications, right? Generally speaking, FISH is very labor intensive. What we're seeing, and this is probably not a problem just in our institution, I think it's systemic, is that labor intensive procedures are being displaced by things that can be a bit more automated. FISH and karyotyping testing in general are taking a lot of hands-on time. So we're looking for ways to replace these techniques because we just don't have enough qualified technologists to be able to do this work as the indications and need for testing expense. So, my full expectation is that more and more of this work is going to be done by kind of integrated molecular-based technologies going forward.
Thank you very much. Thanks. I think makes absolutely a lot of sense. Gary, actually two questions for you. I'll try to combine them. The question is about if you could elaborate a little bit more about the way that you extract total nucleic acid. And as a second follow-up question, I'll try to stick them together. You've shown some very early but still quite solid preliminary data on the fully automated solution that you're testing. When you mean fully automated, do you also mean that this is going from samples to result, including the extraction process?
Yeah, great couple of questions in that, Lucas. So first, let's go with what I presented on the semi-automated system, the commercial GeneStrat NGS test, which we've published the methods, by the way. So if anyone's interested in the detail of extraction, it is not automated. It is manual. We do ship using a STREC, cfDNA tube. I think I can say that. It's pretty common in the liquid biopsy field. and extract using a manual kit. So much aligned with our GeneStrat ddPCR product is the NGS test. Now that makes sense because sometimes there's a reflex and a need to test by ddPCR our NGS test results for confirmation, things like that. So that extraction process is actually the same. Now, on the second part of the question regarding the fully automated system. So that system does include an extraction module. However, in the studies I shared, it was still remnant materials from the manual extraction. We're looking forward to integrating the automated purification of nucleic acids, tissue, and plasma into that fully automated work stream. So great questions.
All right. Well, thank you very much for sharing some additional details in here. So I have a final question for both of you. And so first one, Bekim. So you talked a little bit about the methylation, the epigenetics, that it's coming through. But maybe from your point of view, what's the next hot thing that you envision the field needs to go when we're talking about heme? What should we be ready for?
Well, I would say, for hematologic oncology, solid tumors, as well as constitutional hereditary genetic testing, we're facing a big problem, and that is that we're able to sequence genomes relatively effectively and quickly and, cost efficiently. But we're, seeing this issue. And the issue is that while we can sequence the entire genomes, we still can't interpret the vast majority of that information. Variants of unknown clinical significance are a huge burden, and in all these indications, including myeloid malignancy, there's a substantial proportion of cases where we see what are the tier 3 variants in many of these genes, because oftentimes they're rare, uncommon changes that we have a very hard time deciphering. So there is this big effort in the field in general in genomics is to kind of move towards functional genomics. We're talking about proteomics, metabolomics, transcriptomics, and epigenomics. I actually think epigenomics is probably going to be the front runner, and I am a little bit biased there because I've been, I spent a good chunk of my lifetime doing research in DNA methylation profiling. But we're seeing DNA methylation, genome-wide DNA methylation, epi-signatures as biomarkers to interpret genetic variation, to help stratify, tumor subtypes, to help diagnose rare disorders, move into clinic rapidly over the last couple of years. I actually think hematologic malignancy is one of these, conditions that really deserves a lot of attention here. And I brought it up because, as you're probably aware, majority of the genes that are involved in subtype in hematologic malignancy or for diagnostic, prognostic, clinical implications have to do with regulation of epigenomes. So if you're able to derive knowledge on consequences of those mutations in these genes, they may reflect themselves in global methylation patterns. For example, DNA methylation probably preferred because it's DNA analyte. It's very stable. It can be scaled and implemented in clinical setting. Then we may actually be on to possibility to use this additional functional genomic layer to further substratify these patient populations, but also to interpret genetic variation. We're really excited about it. We've seen this, as I said, move in many different indications, and I do think next few years there'll be some really interesting stuff coming up in heme.
Well, thank you very much, Bekim. I would say I couldn't agree with you more, and I'm very much looking forward, honestly, to get some more data around that. Now, the very last question for Gary. Gary, congratulations for everything that Biodesic has been done in the last few years, all the achievement, the expansion. But the question from the audience is, we understand, you're testing now new solution, including the fully automated one. But the question is, in case you can share, of course, I understand you might not want or not be in the position of sharing that. But what is coming next for Biodesic? So do you plan to expand your portfolio? Do you plan to expand in general operation? If you could tell us a little bit more about what an outlook about the Biodesic plan.
Well, I think again, as a publicly traded company, you're mentioning it, Luca, I've got to be just a little careful with what's already in the public domain. But I think it's pretty clear where we're focused. Late stage targeted therapies, work as I presented today, but also very, very much in early disease. And I'm very happy you had Bekim here also talking about things like MRD in hematologic malignancies, but also, addressing that continuum of care, our focus being lung cancer, lung diseases, looking at nodules, looking at early cancer, our Notify product, all the way through the GeneStrat Targeted Therapies NGS solution. So I think, broadly expanding on those indications, applying the technologies that are relevant for sensitive and specific measurement of disease targets is where we're at.
Thank you very much. Appreciate it. It was a challenging question. I apologize for that. I know, but we want to try to make sure, we pick as many requests from the audience as possible. So with that, I would like to thank you both once again for your presentation, for this very intriguing discussion, and again, for accepting our invitation at this Oncomind World. With this, we'll now taking a break, and we will reconvene here in about 15 minutes for the very last part of this Oncomind World. Once again, thank you for being here with us.
Thank you.
Thank you.